Daniel is an assistant professor in the Department of Epidemiology and the Robert Butler Columbia Aging Center. He is at the forefront of integrating population health with genomics. And his passion is really understanding interventions. So he shares a lot of interest with our group. He is studying the effects of caloric restriction and moving into interventions on social mobility, socioeconomic status. So we'll hear about that at the end. One thing I really appreciate about Daniel's work is his deep curiosity for both the different mathematical models and ways of looking at aging algorithms and comparing them. So it's this, each paper feels like this heavy lift where he's looking at often at many different ways of looking at biological aging. And then his range of measures is completely agnostic. So if I want an objective opinion about, comparative markers of aging and how well they predict mortality or work in an intervention, I look at his papers. He's absolutely prolific. So welcome, Daniel. Thank you.
Okay, well, I'm going to see if I can keep track of two things at once. I already broke a piece of furniture today, so it's hopefully smooth sailing from here. Thanks, everyone, for coming out and giving me the opportunity to talk with you, and to those of you who are tuning in online. This talk hopefully provides some rehearsal for those of you who caught Terry Moffat's talk yesterday, and some new flavor beyond what you heard from Steve earlier. I think it's a little daunting to be referred to as a pioneer following Steve Horvath, but I'll do my best to make a contribution and at least keep you entertained while I'm up here. This is a disclosure. These are my collaborators, and I want to take a minute before I get started to acknowledge them. Terry Moffat and Avshalom Caspi, my mentors for many years, now collaborators. David Corcoran recently moved to UNC, Karen Sugden at Duke, Richie Poulton at the University of Otago in New Zealand, all of whom worked to develop this DunedinPACE measure that I'll tell you a little bit about farther down in the talk. Andrea Baccarelli and Zhu Gao at Columbia. Zhu is now at Peking University. Michael Cobour and David Lin at the University of British Columbia. Bill Krause, Virginia Krause, and Kim Huffman, my collaborators on the CALERIE study, which I'll tell you about toward the end of the talk, and then the various foundations and institutes that have supported the work, especially the NIA. Here's the outline of what I'm going to try and get through in my 40 minutes today. And there really are just 3 things, but I've given you 6 bullets, and I'll move through them and mark them off as I progress. What I'm going to start with, and you've now heard this at least twice already, perhaps 3 times. So by the end, you should be a pro. Geroscience, geroprotectors, and what we're doing trying to measure this biological age construct anyway. We begin with the idea that aging can be understood as a biological process. It's a process of growth and development in the minds of humans as they age, but in their bodies, we can see it as a process of the gradual loss of system integrity with advancing age. And in the past 20 or 30 years, there's been a crystallization of knowledge within the biology of aging that loss of system integrity begins with and is driven by the accumulation of a series of cellular level changes, sometimes called hallmarks or pillars. Steve spoke about them in his talk. You can see them arrayed at the bottom of this slide. We talked about telomere attrition, cellular senescence, mitochondrial dysregulation, loss of proteostasis, and so forth. I'm not going to delve too deeply into the basic biology of aging. My effort here is an attempt to translate that biology into a new set of metrics that can help us advance intervention to prolong healthy lifespan. So with that in mind, let me offer you my version of the geroscience hypothesis, or it's really just my cartoon of the geroscience hypothesis. This is a model in which this accumulation of hallmarks or pillars of aging within and between ourselves feeds forward to drive a loss of integrity and an undermining of resilience in our organ systems, ultimately feeding forward to drive decline in our bodily functions, causing disease and death. And the geroscience hypothesis is that intervention to slow or reverse these molecular hallmarks can delay or prevent the loss of system integrity and thereby extend the period of life lived free of disease. And there are already, as I think Steve referenced, as Terry referenced, a number of therapies that have passed proof of concept in animal models. And these range from the relatively pedestrian, the diabetes drug metformin is perhaps the leading candidate for human translation as a drug to slow processes of aging and extend health span. Repurposed cancer drugs designed to clear senescent cells from diseased tissues. These are senolytics. Perhaps a little bit more exotic, the drug rapamycin, which Steve referenced. It's represented here by the Easter Island monolith because rapamycin was derived from a bacterium discovered in the soil of Rapa Nui, and we now give it to transplant patients to prevent rejection of new organs. It also appears to have geroprotective properties. But beyond these medicines, there are also social and behavioral interventions, exercise, perhaps the closest thing to a fountain of youth that we have, calorie restriction, I'll talk to you about later on, and even positive social engagement may have potential to alter hallmarks of aging and prolong healthy lifespan. So if you are a C. elegans worm, a fruit fly, a C57 black 6 mouse, this is a Sprague Dolly rat, I think, we can slow down your aging. We can give you more years of healthy life. I don't think anybody in the room is one of those things, and we are still working on solving that problem in humans. And as Terry articulated yesterday, the bottom line challenge we have in attempting to translate these very promising therapies from model organisms to humans is that we live an awfully long time. And the latency between the development of disease processes arising from this aging-related accumulation of cellular change driving loss of system integrity and the onset of disease is long. We would need to be running clinical trials lasting decades, and we need to be running trials that last only years. And this need for a short-term measurement of the long-term effects of these interventions on health span has driven efforts to develop metrics of biological age. And in the figure, this construct is represented by chronological age on the x-axis, biological age, the accumulation of damage in the system or the loss of integrity on the y-axis, and the figure illustrating that at any given time in the human age span, there may be a variety of biological ages observed. So everybody who is 50 is not 50 on the inside. And the idea behind one of these metrics of biological age is that we could deliver an intervention and then track the progress of biological aging in the treatment and control groups to test the hypothesis that our treated group is gaining biological age at a slower pace than our control group. And if we're even more ambitious, we might imagine that the slope of that blue line would point down and there would be a rejuvenation process. And again, in animals, that appears to be possible. Okay, so with the preliminaries out of the way then, how do we measure this thing? So what I'm going to do here is give a broad conceptual overview echoing many of the themes that Steve introduced in his talk, and then I'll get on to the work that I've been up to for the last few years. So I want to first acknowledge in the beginning that this idea of surrogate endpoints for health span extension is not at all a new one. There's been work for, I think, quite a while, and I'll call this hypothesis-driven quantification of biological aging, to sum up the accumulation of deficits or the emergence of frailty in humans by counting up disabilities, diseases, or systems that exhibit dysregulation. And there are a list of a few well-known metrics that I think fall into this broad category. But with the figure, which I believe is in the upper right of your screen, I suppose they're the same, isn't that nice? Is highlighting is that these metrics are oriented toward the second half of that geroscience cartoon. They are the evidence of the loss of system integrity, not the precursors to it. And as a result, they tend to have relatively low variability in younger people. They don't reveal risk until it's already established. And because they represent this more advanced stage in the development of aging-related disease, they may be less sensitive to interventions seeking to change them. What has emerged in the last decade or so are a family of what I'll call data-driven approaches that aim to devise more sensitive indices. And the rough idea of these many approaches, and I think Steve is perhaps the progenitor of many of them, and certainly gave us examples of several, are to model surrogates of this latent construct of biological aging from big data, to devise prediction algorithms that can deliver us proxy measures of what is ultimately this unobserved process of the accumulation of hallmarks of aging. So I'm going to talk next about the kinds of data that are getting used and the sorts of surrogates that are being applied. So again, within the cartoon model, we have deficit accumulation and frailty on the far side. We have this loss of system integrity, which we can proxy through measurements of organs and organ systems. This is organ function testing, blood pressure, lung function, cardiorespiratory fitness, measures of body composition, and blood chemistries, complete blood counts, those kinds of data. Taking a further step backward in this chain of causation, it may be possible to directly measure cellular level changes that provide readouts on the accumulation of pillars or hallmarks of aging, including from DNA methylation data, gene expression data, and more recently proteomic, metabolomic data. And that, et cetera, is doing a lot of work for me here because there's a whole family of omics technologies that are emerging and that in their integration may give us new views on biological aging. So what kind of surrogates do we think about when we're doing this data mining to uncover signs of a biological process of aging? The first and perhaps still the most widely used is time since birth. And this is how many of the original epigenetic clocks were developed. And they simply compare older to younger individuals and characterize aging as molecular differences between old and young. A more recently developed approach is to focus attention on the time remaining in life. And here, these are models based on mortality, or as Steve mentioned, longevity, to again identify molecular correlates of shorter and longer lifespans. And then finally, perhaps a less well-studied approach that my colleague Alan Cohen has been particularly aggressive in pursuing is measures of loss of coordination among biological systems. And this is a sort of a direct descendant of Linda Fried's original frailty construct and an effort to translate that into this data-driven approach that may be able to provide us an early window on the emergence of frailty in younger people. So these data-driven approaches have the advantage of showing more variability in younger people, the potential to provide these preclinical indications of risk to detect processes of aging that may drive disease before the signs of those diseases are clinically observable. And because they're farther up the chain, they may be more sensitive to our interventions. They may give us a better opportunity to detect when a therapy aiming to slow or reverse accumulation of molecular hallmarks is having the desired effect. There's been a great deal of progress in perhaps the past decade in the development of these indices. Most pronounced, I think, at least in my view, is the dramatic improvement in the accuracy of prediction of health endpoints. So particularly with the transition from clocks based on chronological age, time since birth, to clocks based on mortality or time until death, we've seen substantial increments in the predictive accuracy for mortality and increasing accuracy for the prediction of incident disease and disability. In parallel, there's been this growing sophistication of methodological approaches to refine these indices from big data sets. Steve mentioned elastic net regression, linear models. There are increasing efforts to move beyond these linear models using deep learning and neural networks. As computing power increases, ever more sophisticated models are being applied to these data. And it remains to be seen whether they will bear fruit, whether we will in fact be able to build a better mousetrap from a more complicated statistical model. But that is an area of progress. And then finally, this increasing diversity and potential for integration across molecular levels of analysis. Studies of aging are increasingly measuring not one, but 2, 3, 4 of these molecular substrates, allowing for the development of aging metrics across levels of analysis and the integration of data across these levels of analysis to devise more refined measures of aging. Those advances are pushing against a series of frontiers. You heard a lot from Steve today about elucidating the biology driving epigenetic clocks. Another frontier is the external validity of these measures, which are still primarily developed and tested in samples that are white, well-educated, and often bio-curious. These are the people who show up at medical centers and provide their samples. Although again, substantial progress is being made on this front. Thirdly, the modifiability of these indices in humans in particular is uncertain. And I'll talk in a minute about why I think the data-driven measures of biological aging that we study in humans are potentially quite different from the measurements that we're looking at in animals. And then finally, there remain challenges with the reporting of study results. I find that in many investigators in the field have now adopted a comparative approach to studying these metrics of aging, where results are reported not for one, but for several of the measures. However, that's far from a universal practice, and you still see papers introducing new metrics of aging in which the sole criterion is correlation with chronological age, and that is the sole validation, which I think leaves us without very much information about what these new measures are actually telling us about this process of human health span. Okay, so that progress, those frontiers aside, what I want to do now is problematize the whole construct of biological age for the purposes of geroscience in service of motivating the introduction of a measure that you got a teaser from Terry Moffat about yesterday, with the caveat that I'm not suggesting at all in this case that biological age isn't useful, but that it may not be a complete or total summary of the information we need to evaluate geroprotective therapies. So there are three limitations to the construct of biological age I want to introduce: mortality selection, cohort effects, and uncertain timing of when differences emerge in development. And I'll talk about each of them in turn. So what I mean by mortality selection here is that old humans and young humans sampled into a research study represent qualitatively different populations. The old humans are by definition survivors. When we recruit 70 and 80-year-olds in the clinic, we are observing only, even if we have a completely representative sample of the surviving population, we're observing only a third to a half of the original birth cohorts. The others have died by that time. So our old representation, what we're using as a surrogate for aging in these studies, in some sense, is weighted to represent the slow agers at the far end of the distribution. So that may attenuate signal when we attempt to refine clocks or other metrics of aging. It's noteworthy, I think, that this particular challenge is substantially mitigated in the metrics of aging devised from studies of mortality, although not entirely, because most of the deaths that occur in our follow-up studies occur to people at the end of life. The second limitation is the cohort effect. In cross-sectional data, how old you are and when you were born are the same thing. And when you were born can be an important measure of the exposure regime to which you've the exposure regime you've experienced across your life. So we can think about the prevalence of smoking in the population as something that's changed substantially over history and therefore differentially affects people born at different times. Lead came out of gasoline. Vaccines were introduced. The quality of prenatal care has changed dramatically. The adequacy of prenatal nutrition and the prevalence of other insults to child development has declined. Finally, and I think this is in some sense the most important of these, we don't know in a measure of biological age when the difference we observe between a person's biological age and their chronological age was established. And data accumulating now, and this is a figure from one of Vadim Gladishev's recent theoretical papers, but they have a lovely piece in Science Advances that was just published, suggests that substantial variation in epigenetic aging may be induced in the very earliest stages of life. Now, that doesn't make these measures any less predictive of outcomes, but it may affect the extent to which we can modify them through interventions delivered to people who are in the middle and later stages of life. Okay, so with an understanding of those potential limitations of the construct of biological age, we've been working for the past 7 or 8 years, I suppose, in Terry's case, perhaps a bit longer, on an alternative approach. Rather than measuring how old people are when we observe them, we're attempting to measure how fast their bodies are changing. So a biological age, whether it's an epigenetic clock or a blood chemistry clock or a metabolomic clock, is essentially a summary of how much aging has occurred in the person up to the time I draw their blood. And as a result, it faces the limitations of mortality selection, cohort effects, and uncertain timing of when the variation was generated. So the alternative is you can measure some pace of aging construct that addresses how fast the person's body is changing. And the theory behind the pace of aging metric is that if aging is characterized by this gradual and progressive loss of integrity across bodily systems, then we can infer the unobserved process by tracking changes occurring in a coordinated fashion in multiple different organ systems. And if this is in fact a lifelong process, then this decline should be observable already by young adulthood. So the original paper that we published back in 2015 looked at 12 years of data. Terry showed you yesterday a more complete follow-up extending through the 45th year of these participants' lives. But essentially what's done here is the Dunedin study's comprehensive measurement of the physiologies of its participants from young adulthood at age 26 through, in this case, their 38th birthday, their cardiovascular system, renal system, hepatic system, pulmonary system, their gums, their immune systems, their DNA as measured by telomere length. It's possible to track the rate of change for each person across these 3 and subsequently 4 time points, and to quantify for each marker how much faster or slower a person is aging than the cohort norm. Those differences can then be composited to form a single index summarizing whether the person is aging fast or slow, their pace of aging. And as Temi explained yesterday, this pace of aging is perhaps important to observe that it does vary in these young people. It is associated with early life risks and does predict later life functional deficits, not much later in the case of these individuals, only through their 45th birthday. But it's very challenging to export from the hothouse environment of a cohort study into clinical trials or population surveys. And with that in mind, we undertook efforts motivated by the work that Steve and his colleagues had done to translate this pace of aging measure into a single time point blood test. And this distillation, I guess is how I've tended to talk about it, of the 12-year pace of aging into this single time point DNA methylation blood test we published last year in eLife, and we called this measure DunedinPAAM for pace of aging in methylation. I have to say that name has not stuck and is not easy for anyone to work with. And with that in mind, we renamed it for the second generation. So DunedinPACE is built on the same principles as the original DunedinPAAM, but takes advantage of this additional wave of follow-up. So there are now 4 repeated measures of, in this case, 19 biomarkers tracked across a nearly 20-year period. And these are then distilled into a blood test through elastic net regression machine learning applied to DNA methylation, with one important change from the original. And this change was driven by an observation that Karen Sugden published last year in Cell Patterns, which was not news to many people working in the DNA methylation space, but which did take us by surprise, which was that the individual CpG sites on Illumina arrays are not terribly reliable measurements. So the graph here shows the frequency of the probes on the y-axis and the test-retest ICC on the x-axis. And what the figure is showing you is that the majority of probes on the Illumina array measured in blood have relatively poor test-retest reliability. Now you can see that the green box plots at the bottom of the figure are showing data for the probes included in the original multi-tissue clock that Steve developed, the Hannum clock, and then Morgan Levine's PhenoAge clock. And you can see that those all include relatively more reliable probes than the array norm, which is what you would expect from a machine learning algorithm that's meant to sift through the data and find the signal. But still relatively low reliability. And as a paper, as a preprint up on bioRxiv from Morgan Levine's lab shows now, the consequence is that the test-retest reliability of most of these epigenetic clocks and our DunedinPAAM measure is not great. It may be in the sort of 0.8 range, which is perhaps fine for a cross-sectional study. But if you want to test in an intervention trial whether you can change a measure, a test-retest reliability of 0.8 is going to seriously harm your statistical power. The GrimAge, it's noteworthy, performs better in this exercise. But with this in mind, when it came time to devise the DunedinPACE algorithm, we focused our machine learning analysis on just the subset of probes on the epic array that showed the highest reliability. So in this case, the threshold we used was a test-retest reliability ICC of 0.4, still not that high, yielding about 80,000 probes fed into the machine learning analysis. And the consequence of this change, we think, it's the consequence, is substantially improved reliability of the measure. So this is a boring detail, but I promise it's going to be important as we get to the end of the talk. You see here, this is a public data set of 36 samples run in adjacent wells on an Illumina 450K array, plotting replicates against each other. And that's the plot that you'd hope to see. What you can see in the figure to the right are data reporting the ICCs in 3 data sets for DunedinPACE, the original DunedinPOAM measure, as well as these benchmark epigenetic clocks. And GrimAge is the one at the far end there, and you can see also shows very high test-retest reliability. Again, the differences between these clocks may seem subtle, but in the context of a randomized trial, depending on measures of change, the difference between 0.8 and 0.95 is very large. Okay, a few other observations about DunedinPACE. This plot shows a correlation of DunedinPACE with chronological age. It's not terribly interesting, and it's certainly much weaker than what you'd expect with any of the epigenetic clocks, but keep in mind, this is not a measure of biological age. This is a measure of the rate of aging. What this plot shows us is that according to this measure, the rate of aging, the pace of aging, is accelerating as we age. And that is consistent with observations made of mortality risks. So in demography, we know that people's risk of death accelerates as they grow older. But this has not been something that could be tested in a biological age space with existing clocks, because when we measure the aging rate from a biological age, we regress chronological age out of it. So it's by construction uncorrelated. Here we have a metric that can evaluate whether this demographic observation is replicated at the, in this case, cellular level. And we have some evidence that it is. People with a faster Dunedin-pace, pace of aging, also have more advanced biological age. Here you can see a relatively high correlation with the GrimAge, more modest correlations with some of the other clocks. And in the paper, we published correlations with some blood chemistry clocks, which are similar to the correlations with PhenoAge and the Hannum clock. DunedinPACE, as Temi shared with you yesterday, is associated with morbidity and mortality. And these data are important because they tell you that people who are aging faster are at increased risk of dying, and as I'll show you next, getting sick. But it's also important because the people in these figures from the normative aging study in that top survival plot and the Framingham Heart study in the bottom survival plot are much older than the Dunedin study participants. So devising a measure of the pace of aging in people who are in their 20s, 30s, and 40s is giving us a metric that can inform us about health span in people who are in their 70s and 80s. And the comparative data are in the plot on the right-hand side of your slide, showing the hazard ratio for mortality on the y-axis, and then listing the several clocks on the x-axis. These are the data on incident and prevalent chronic disease from the normative aging study. So I think of particular interest are the data on the left-hand side of this figure, showing that among these old men, they are older, they were all 71 or older at the time their blood was drawn. Having a faster Dunedin pace predicts an increased risk of developing a new chronic disease. And these are data that Temi again showed yesterday on dementia risk. So you can see that the data for cognitively normal participants are plotted in blue, those with mild cognitive impairment in orange, and those with frank dementia in gray. And the data for DunedinPACE are on the far right of the slide. GrimAge data are adjacent, and then the other clocks to their left. Finally, and this is particularly important to the work that I'm involved in, DunedinPACE indicates faster aging in young people exposed to early life adversity. These are data from the E-RISK longitudinal study, another of Terry Moffat's cohorts. These are about 1,000 18-year-olds who gave blood and for whom prospective records had been collected of their family socioeconomic status. These are from age 5, and then of their victimization by peers and adults through age 12. And what we see in these data is that kids who grew up poor are aging faster. The Cohen's D comparing the low socioeconomic status to high socioeconomic status kids is about 0.4, which is a sizable effect for a biomarker. And the effect size for childhood victimization is about 0.5. Okay. So I have about 10 minutes left, and I want to share with you now what these measures, in some sense, are designed for, which is, can we use them to detect the effect of an intervention? So the setting for this study is the CALERIE trial. CALERIE is a randomized controlled trial of calorie restriction run by the National Institute on Aging between 2007 and 2009. Calorie restriction, for those of you who are not familiar, is perhaps the best established geroprotective intervention in animals. We've known since the early 20th century that reducing macronutrient intake with maintenance of micronutrient sufficiency in mice extends their lifespan. And since that time, the finding has generalized to a range of other species, flies, worms, and in data from the NIA, perhaps even rhesus monkeys, although what we know from the monkeys is that calorie restriction extends health span. The data on lifespan are still coming in. So we don't know the answer to that question yet. But these data were sufficiently promising to motivate NIA to randomize 220 non-obese adults to 25% calorie restriction or regular diet for a period of 2 years. Now, calorie restriction is really hard. And 25% CR, if you're a mouse, is not that much. Mouse CR experiments often use a much more significant dose. But in humans, this was really a challenge. And if you look at the box plots, what I'm showing you there are percent calorie restriction measured by doubly labeled water for the ad libitum group, the regular diet group, the control group on the left. And that's the 2 boxes. And then the cluster of 4 boxes on the right are showing you calorie restriction at 6, 12, 18, and 24 months in the treatment group on the right-hand side. And if you can't quite squint to see the y-axis, what it's showing you is that on average in the treatment group, people were barely getting to 20% at 6 months. And the dose they achieved attenuated from there to about half of what was prescribed by the end of the trial. Nevertheless, calorie-induced significant and sustained weight loss. So that's what the far right-hand side figure on your slide is showing you. They lost on average about 8 kilograms, and that weight was stable then through the end of the trial. There have been a number of follow-ups showing that calorie improved indices of cardiometabolic health. We published a paper in 2017 here, more recent data from a preprint that Dae-Eun Kwon has put up, showing that blood chemistry measures of biological age show that the rate of increase was slowed by calorie intervention. But a limitation of these blood chemistry analyses of calorie is that the intervention acts directly on the organ systems producing the biomarkers that are used to quantify aging in this case. And so with that in mind, we asked the NIA, and were granted funding, to conduct a molecular analysis of the treatment effects in calorie. And so what we did here was we took the blood samples, or the DNA that had been extracted from those blood samples from the baseline pre-intervention, the 12-month follow-up, and the 24-month follow-up, and we ran Illumina epic arrays and computed several indices of biological aging. And this is what we found for 4 of the epigenetic clocks. So you see the original multi-tissue clock in the top left, the clock proposed by Hahnemann colleagues in the top right, and then in the middle row, these 2, what Steve called second-generation clocks. So these were built to predict mortality, PhenoAge on the left, GrimAge on the right. And for 3 of these clocks, for the first-generation clocks and for PhenoAge, we actually see the opposite pattern from what we would expect. There's no substantive difference in the rate of increase. So I should explain, the y-axis here is the increase in clock age from baseline. So it's 0 at baseline, and then we expect it to be 1 at 12 months and 2 at 24 months. And further, per the hypothesis that calorie restriction should slow aging, we expect the red line for the calorie restriction group to sit below the blue line for the ad libitum control group. And we do see that for GrimAge. We don't see that for the other 3 clocks. So for GrimAge, at 12 months, we see a hint of a geroprotective effect, but by 24 months, the treatment group and the control group look similar. The final row of data here are for these pace of aging metrics. And in this case, the y-axis has a different interpretation. We're still quantifying change from baseline. But in this case, the change from baseline represents a change in what we're trying to measure, at least, to be the pace of aging. So as the red line dips below 0, that's telling us that the intervention has slowed the pace of aging. And what we see for both the first generation DunedinPAAM and then the second generation DunedinPACE is that the pace of aging is slowed from baseline to 12-month follow-up, and that slowed pace of aging is then maintained through 24 months of follow-up. In the case of DunedinPAAM, we see there's no difference between treatment and control at the second follow-up. There's the hint of a difference at this first follow-up. I'm going to translate these differences now into effect sizes to make them a little easier to look at. This is the intent to treat effect on the y-axis, so just a simple comparison of participants based on their random assignment. What you see are the 12 and 24-month data plotted adjacent to one another for each of these 6 measures of aging. And I've drawn a line in the middle of the chart here just to separate those clocks that show evidence of slowed aging in caloric restriction from those that are not showing that effect. And you can see the horizontal line intersecting the graph here is 0. So as you saw in the plots of change, if anything, there is this net positive effect on these the first generation epigenetic clocks and perhaps the phenoage, whereas for GrimAge and the pace of aging measures, we see evidence of a potential geroprotective effect. But it's only for this most recent DunedinPACE measure that we see consistent and statistically significant differences in the treatment group as compared to the control group. And so the question is, like, why should that be? So one of the reasons is the improved reliability of the measurement. I'm going to flip forward and then I'll flip back and then I'll wrap up. These plots show you the intent to treat effect size on the y-axis plotted against the test-retest reliability from that publicly available 36 sample DNA replicate data set on the x-axis. And so as you move from left to right, you're going from low reliability to high reliability. And as you move from the top to the bottom of the graph, you're seeing increasing geroprotective effects. And what we see here is that the more reliable measures show stronger evidence of a geroprotective effect. Nevertheless, DunedinPACE and GrimAge have fairly similar reliabilities in these data sets. There's a difference in effect size. Is this random noise? Perhaps. But I think it's also possible that the difference in results we see for the DunedinPACE measure and the pace of aging measures more generally may reflect differential sensitivity to intervention having to do with the way these measurements were designed. So if you measure the pace of aging instead of the state of aging, you may have a quantity more sensitive to intervention, particularly when that intervention doesn't, in fact, rejuvenate the organism. It's not dialing back the clock. It's simply slowing down its ticking rate. If you think about the information contained in GrimAge, for example, it's summarizing the increase in aging that has accumulated over the entire lifespan. So the portion of that can be reversed by 2 years of calorie restriction may be relatively modest, and therefore there may be differential power to detect a more subtle intervention effect. It could also be that if we ran this experiment again and drew different blood samples, we'd get a different result. I think it's noteworthy. There was a paper recently published on a calorie restriction intervention in an obese sample that did show an effect on the GrimAge. So more to come. So finally, I'll wrap up. I'll show a few more pictures and then we can have questions. But what I want to leave you with is this idea that methods to quantify processes of biological aging represent new tools for aging science, including our DunedinPACE measure as a new addition to this armamentarium that critically is conceptually distinct from the clocks. And we think, and we hope, complementary to them in understanding differences in human aging and the effects of our interventions. And I want to encourage, and you'll see much more of this in the session to come, the audience here to consider taking up these measures into your own studies. Much of what we need to know about how environments modify the pace and state of biological aging depends on the integration of these measures into social and behavioral sciences. So that's the people in this room. A few examples of the direction we're heading with this. This is a paper now in press at the American Journal of Epidemiology, but you can find it on medRxiv, showing that the first generation DunedinPAM, as well as the GrimAge and these blood chemistry clocks, are predictive of morbidity and mortality in Black and white older adults, and more importantly, mediate substantial fractions of Black-white health disparities in healthy aging. So this is a metric that may give us a readout on interventions designed to ameliorate health disparities in advance of testing changes in disease incidence or differences in lifespan. And the mediation data are in the bar chart at the bottom right of the graph. And you can see significant mediation of differences in short-term mortality risk, in the incidence of disability, less so changes in self-rated health. The question of social mobility came up yesterday. This is a paper that we're getting ready to submit, looking at childhood to later life socioeconomic mobility, again, using data from the Health and Retirement Study. And the interesting finding in this paper, we went to these data to look at the question of whether social mobility was a net benefit or a net cost to individuals on the basis of recently published data. In fact, they go back to the 1970s of the work of Sherman James, suggesting that climbing the social ladder, because it involves crossing social boundaries, may be biologically costly. And we can think about this as sort of an echo to Dr. Thayer's talk of yesterday. Are the benefits of social mobility purely in the economic resources that people have, or do they extend to something like biological aging? What we found in this study was that if we measure social mobility purely from educational data, we can reproduce the effect observed in several other studies, which is that the benefits of upward mobility are much greater for white as compared to Black Americans. But when we include in our measures of mobility the successful accumulation of wealth across the life course, the benefits to Black and white Americans are essentially identical, suggesting that structural barriers to the translation of education into resources, social status, and comfort for Black Americans may impair the development of the benefits to healthy aging that we associate with this kind of upward mobility. So it's not that education isn't effective, it's that education, when it confronts, let's say, a racist society, is insufficient. It's only when it translates into actual resources that we get this amelioration of health disparities. And then finally, I know many of you work with young people and may work with tissues other than blood. These are data that we published in pediatrics earlier this year. That's Laurel Raffington, who's now starting her own lab at Max Planck in Berlin, correlating this first generation DunedinPAM measure with household socioeconomic status in 600 children measured as part of the Texas Twin Project. So we're essentially reproducing the association I showed you in the E-risk study in a younger group of people using saliva DNA. And there's some interesting data in the supplement of that paper on the correspondence of this particular measurement in blood and saliva, suggesting that that kind of translation is a reasonable thing to do. So these slides will be available to get the links, but this is David Corcoran, who I want to credit with writing the software package that actually computes this measure and also the forthcoming one. And for those of you who have blood chemistry measures, blood chemistry data, and are interested in exploring blood chemistry measures of biological aging, Teun has recently written an R package that will allow you to project published algorithms onto your own data. And if you don't have quite the same biomarkers as the originally published papers, to customize those algorithms to the biomarkers that are available in your data set, and in fact compare the performance. As Alyssa said, I'm very interested in comparative validity here. Compare the performance of your new custom algorithm to the originals in the National Health and Nutrition Examination Survey's data to get a sense of whether you're actually getting a true measure of biological age when you modify the algorithm. So thank you to Alyssa, thank you to all of you, and look forward to your questions.
Please get in line. Thank you, Dan. Beautiful.
Sort of ominous to have all these people approaching you in a mask. I don't think I appreciate what that looked like from the stage. But
Hi, I'm a bit of an outsider here, so I apologize if this is a naive question. But oh, my name is Aaron Fisher. I'm at UC Berkeley. I'm just listening to these last two talks and these calculations of these clocks, and I'm wondering how familiar folks here are with the ergodic. Oh, they're cutting me off. And the idea that the premise that between subject variance, covariance, and within subject variance, covariance are not necessarily related, because these clocks are being built by between subjects data, but of course aging is a within subject process. And I'm thinking about, the metrics you're giving us, and about 34% of the variance between your predictor and biological age, and you mentioned a Cohen's d of 0.4, which is just 4/10 of a standard deviation. And sometimes we statistically applaud ourselves, but the idea of the actual size of these effects are pretty small. And so I'm just wondering if there's any attempts to train these clocks, to build these clocks at the within subject level, and the promise that might have to actually give a little more predictive punch, since that's the level that you're really trying to understand.
So that's a good question. That's what we did.
The clocks are trained within subject?
So the DunedinPACE and DunedinPOAM measures are based on this pace of aging construct, which is itself a within person measure. So the same people were measured when they were 26, 32, 38, and finally when they were 45 years old. And the same measurements were taken at all of those time points. So the first step in each of these models is to fit a series of growth models to those within subjects data, and to predict for each person how much faster or slower that biomarker changed for them relative to the cohort norm.
Right.
It's the sex specific.
Well, I don't want to take up too much time. I'll grab you somewhere else sometime else. So growth models are still between subject covariance models, and I'm talking about more of a time series based approach. But yeah, because 5 observations, you can't actually converge a statistical model around 5 observations. So you would need 50, 100, 150, 200,
I see what you're saying.
300,
You're fitting individual specific models for each.
Exactly. And then using that level of covariance. But
I think that's a tremendously exciting future direction. And if you give us another 50 years and $50 million, we'll come back to you with the results. But it may be possible as we move toward more universal collection of these kinds of data to approximate something like that. So interesting idea.
Yeah.
Very cool. Thank you, Dan. We have a lot of good questions online. So I apologize if I can't get to all of them. Maybe we can ask Dan to take a look at them and answer some, if I could wrangle you in. But I'm going to ask a question from Richard Lane. An index of aging is decreased coordination between organ systems. Has anyone attempted to relate this change in coordination to structural or functional brain measures?
That is a really good question. I brought up, now I'm going all the way back to the beginning of the presentation, Alan Cohen's work on homeostatic dysregulation, which is a Mahalanobis distance-based measure of biological aging. To my knowledge, Mahalanobis distance-based and PCA-based measures of aging, this coordination-focused approach, have not been correlated with MRI data. But I will let Alan know that that is an interest, and I'm sure Alan is eager to get into brains when he can.
Okay, I will let the next person ask.
I think that, there we are. Sorry about that. I think that these indices of cross-system coordination are conceptually extremely appealing. I think they've been less studied than have other measures of aging. I know that in CALERIE, for example, we see weaker effects for the statistical distance-based measures than we do for the other measures, but I think it's fair to argue that we've not implemented them with perfect fidelity. So I think this is a good idea in search of more rigorous validation. I would say that they have the distinct advantage of not enforcing unidirectional models on the way biomarkers relate to aging. And so I think what motivated Alan in part was this concern that, high CRP is bad, but super low CRP might not be very good either. And in fact, what may matter more is when, for example, your various cytokines are out of balance with one another, rather than when they're simply up or down. And that's an idea that immunologists are getting excited about. Eric.
Hey, great talk. And honestly, I think we can all agree, a great presentation style. It was very entertaining. I loved it. I did have a question though, because I've been really interested in these methods, but there's always been a concern about the the training of them in the homogeneity of the sample, right? And so, you know, there was in the early clocks, there were kind of racial differences that emerged, but they were largely a function of kind of the people that were included in the trials that then were developed. And, you know, and then there was, you know, the issue of like, how do we do this with children and kind of, and those samples. And so, you know, in the Dunedin sample, right, it's a pretty uniform population. And the idea of using it for interventions raises to me the concern that you may interpret differences that are a function of the sample and less a function, you know, where it will look like it doesn't work for one sample, but because of the heterogeneity of the population, like in low SES populations or racial differences. And I know that you're finding some racial differences, but I'm just wondering if you could speak to how the actual sample that it was trained on might play a role.
Yeah, I mean, I think maybe we should get Steve up here to comment on some of the epigenetic clock findings as well. But I just want to point out, I think this is a critically important question. And it's now something that I think more teams are attacking in their work. So this paper, where we're looking at mediation, isn't considering only are there differences, but do we in fact see parallel magnitudes of association between differences in aging measures and differences in disease risk between population groups? And in fact, this paper was a nearly 2-year odyssey that Gloria went on, following some results that we published in a sample of older adults from the Duke EPIs, in which we observed really different effect sizes for blood chemistry clocks in older white and older Black adults. And there are a number of reasons we might see those different effect sizes, but we were very concerned with addressing this potential population specificity of the measures. And that may be amplified as we move to genomic measures, where population differences in the frequencies of alleles could lead to artifacts in genomic data that essentially have nothing to do with biology, but which do contaminate our measurements. And at least in this study, what we found was that for the PhenoAge, for the GrimAge, and for DunedinPAM, effect sizes are pretty consistent between Black and white older adults. And where we do see differences, what they essentially reflect is that even for people who are, the base rate of disease and disability in older Black Americans, because it's already elevated relative to white Americans, constrains how much of a gradient you can observe. So the presence of health disparities across the population and the carrying capacity of the organism for a certain burden of morbidity and disability before senescence is going to produce perhaps small differences in effect sizes between groups, but the basic criterion validity of the measures appears to be preserved. But I think that this is an important question and one that needs more attention. Steve, did you have anything you might want to add in terms of ancestry and epigenetic aging?
Sorry, the original clocks were built on different racial groups. You know, they apply to all racial groups. We often see an offset. It depends a bit on the clock, and yeah, I'll stop with that.
Fantastic research. I'm Shaoyun Su from Medical College of Georgia, Augusta, Georgia. So thanks so much. Answer the question about the Black-white health disparity. My question is also related to that. Any studies can validate the pace of aging markers in the African American population? And the second question is, how about the sex effect? We know men and women have a different aging rate, and should we construct the pace of aging separately in men and women, or we can use it?
So let me take your second question first about men and women. So interestingly, the pace of aging itself is modeled essentially separately in men and women. We allow the slopes to be different for each biomarker in men and women. They're not that different in the Dunedin study in this age range. But when we distill that longitudinal pace of aging measurement in the Dunedin study, which by construction shows no sex differences, into a DNA methylation metric, and we take it into other studies, what we see, consistent with what's been reported for the GrimAge, for PhenoAge, and I believe for the multi-tissue clock and the Hannum clock, is faster aging in men as compared to women, consistent with differences in life expectancy. So I think there are interesting questions about whether those differences are artifacts of kind of the fundamental biology of sex, or differences in risk factors, but that's an open question. To the extent of, is there data validating pace of aging in non-white populations? This paper that we worked on was specifically concerned with that question. So do we see consistent criterion validity, prediction of disability and mortality across race ethnic groups? And the answer in this study is yes. But as Temi mentioned, there are efforts ongoing to bring many more data sets to the table in which to address this question. And it may be the case that this particular population of about 1,000 Black older adults in the health and retirement study whose blood was analyzed for DNA methylation is not representative of the full US population. And in fact, we have strong priors that that's the case, because we know that, well, let me come back to, perhaps in a private conversation, what many of the challenges are when we focus on studying health disparities exclusively in older people. But one of the problems that we face is that if people are dying as a function of their rate of aging, then if we wait to sample people until they're already old, we'll be missing a lot of them. And if we have a population like Black Americans who are dying at younger ages than white people, then we have differential selection into those populations. And so we may essentially be selecting for a more homogeneous cross-ancestry sample in the HRS than we might be otherwise. Now, that's not going to affect the sort of basic biology of aging, but it could have something to do with the exposure histories that people are bringing with them into the study.
Hi, so I noticed in your chart for the CALERIE study that the rate of aging seemed to creep back up once the participants went to 12% versus the 20% calorie restriction. So I'm wondering if you think that such interventions that are shown to reduce the rate of aging are sustainable if someone only, say, does calorie restriction for a year. Are they kind of, resetting their clock and now they're sustaining that new clock, or do they go back to their original clock?
So thank you. This is a great question. We don't know what the long-term effects of CALERIE are going to be, whether a temporary slowing of aging is going to have lasting consequences for health. But SciDOS has been funded by the NIA to follow these folks up 10 years out from the end of the intervention. So we'll find out whether their rate of aging snap back to what we see in the control group, or if it remains slowed, and either way, whether there may be any long-term effects of those differences in the rate of aging on health. But I also want to use your question as an excuse to show one of my backup slides, which is essentially a dose response analysis that we did in CALERIE, where we used instrumental variable regression to try and recover what the effect would have been if we had achieved, in this case, a 20% dose of CR. We didn't go the whole way up to 25 because there just wasn't much data on what 25% CR looked like in CALERIE. But basically, we see the expected magnitude of increase. The effect is a little less than twice as big if we up the dose of calorie restriction. But I should also just clarify, I don't think calorie restriction is a public health intervention. We're not going to scale that to the population, but if we can establish the effects of this very intensive intervention, it'll give us a benchmark against which to evaluate alternative therapies that we may be able to scale. And those could include related therapies like intermittent fasting, or it could be something entirely different. Like, for example, the effects of poverty mitigation experiments that have been funded by the federal government, in which we're working to generate these kinds of aging measures so we can find out if making people's lives better can actually make them age more slowly. So, Nikki.
I hate to interrupt you after that awesome comment. We should all dwell on that for a moment. Quick question. Thank you for referencing the child buccal saliva method. As a child-focused person, can you speak a little bit about how young you think this is useful? Is there enough variability very early in life? Do you have any quick thoughts? I know we don't have a lot of time.
I think Steve is probably in a better position to comment on this than I am. I know that his original multi-tissue clock takes the whole way back to conception. You have negative ages in fetal tissue. With the pace of aging, we don't know. What we see in the kids in that study who ranged in age, I think from 6 to 17, was a normal distribution. And on average, a slower rate of aging than what we observed in, for example, the midlife Dunedin sample where the measure was developed, which is consistent with what we should expect. But whether you could, for example, measure this thing in 2-year-olds and it would be interpretable, I don't know. And I think there's probably a bigger conversation we want to have about what it means to measure aging in very young people. So the discussion last night, I think, included elements of what I would think of as aging, which is this process of a loss of system integrity, of deterioration, but also of accelerated development, which to those of us who study the end of life is not the same thing. They're related. We might expect that a more rapid pace of development could feed forward to drive accelerated deterioration, but those are conceptually distinct parts of our biology. And in fact, the reason aging science and aging biology was so contested for so long was this idea that development was a program and aging was just noise. And so any suggestion that there was systematicity to it was met with a lot of skepticism. But
So much, Daniel. Beautiful. So we have a 15-minute break until our grand finale aging panel. And Dan,